In this video, I'll be explaining Table 1 in the scientific article turning a riboflavin-binding protein into a self-sufficient monooxygenase by cofactor redesign. Monooxygenases are enzymes that catalyze organic substrates. These reactions are very expensive to undergo, so scientists have been working on a way to either circumvent these reactions or make them cheaper. One way to do this is alter what's known as a cofactor, which helps the enzyme speed up its reaction. The authors took a riboflavin-binding protein and altered its cofactor, called a flavin. They took these flavins and altered them in an attempt to boost the reaction output. The product of this reaction is an oxidized sulfide. The results of this experiment were put into this table. In the first column, we have the trial numbers. And in the second column, labeled R1, we have the different organic substrates used in these trials. For example, in trial 2, we used para-toluidine. R2, the third column, labeled R2, refers to the specific R groups attached to these substrates. So in trial 2, that would be a methyl group. In column 4, which is labeled flavin, this talks about the different flavin derivatives used in each trial. So in trial 2, flavin 2 was used. In the column labeled protein, this is talking about either the presence or absence of a protein in this reaction. In this column, labeled conversion, this talks about the sulfide converted in the reaction. And in this last column, these numbers refer to the excess, how much sulfoxides were in excess. The higher these two columns of numbers, the greater the reaction output. And as you can see from the final 3 trials, these do not quite fit the pattern of the first 16. This is because these trials are controls. Trial 17 does not have the protein, and so that the conversion number is going to be slightly less than the rest. In trial 18, there was no flavin cofactor, as you can see, so this number is going to be significantly less than the rest because the reaction was not speeded up through the presence of flavin. In trial 19, the original unaltered flavin was used, and as a result, the conversion of sulfide was again lowered. These 3 trials are used to compare to the altered flavins to show the difference between the unaltered reactions and the reactions that are sped up through the use of an altered cofactor. This table shows that this reaction output can be greatly increased by altering the cofactor flavin.